Hello everyone. Today we are going to study about biological nitrogen fixation, where in which I have divided this video actually into three different parts, where in first part in this particular video we are going to study in general about this nitrogen fixation and the different types of nitrogen fixation and the different microorganisms which are involved in nitrogen fixation, and taking the example of Rhizobium, how the nodule formation happen, the different steps involved in nodule formation we are going to study in this particular video lecture. So before going in detail about the biological nitrogen fixation, let us study what is this nitrogen. As we all know, this is one of the gas which is present in an atmosphere, which constitute about 78% of the total atmospheric gas. As we could see in this pie chart, compared to the other gases which are present in our atmosphere, 78% is constituted by nitrogen. Nitrogen is one of the gas or the element which has got two nitrogen atom which is being bounded by a triple bond, because of which this nitrogen is a inert molecule. So this is the reason also this nitrogen cannot be assimilated by any organism. Even the plants cannot assimilate them as such as the carbon dioxide or oxygen, however it has been assimilated. Because of its inert property, this nitrogen cannot be utilized by the plants or any other microorganism, including animals, excluding some of the prokaryotic microorganism has the ability of fixing such atmospheric nitrogen. However, despite this, nitrogen cannot be utilized by most of the organism. However, this nitrogen is one of the important substance or the element which is required for all the organisms which are present on this earth, because nitrogen is one of the important constituent of a basic genetic material. Say for example, the nucleic acids, DNA, RNA, they have been made up of the nitrogenous basis. Without this nitrogenous basis, no DNA. Without DNA, no proteins. Proteins are the building blocks of all the living organism, and also most all the enzymes have been made up of these proteins. So and in turn, these proteins are also made up of amino acids, where these amino acids are made up of the nitrogen. Apart from all this, the plants, where we are dependent, we are the consumers, where we have dependent on the producer plants. They synthesize their own food by the process of photosynthesis. For the process of photosynthesis, chlorophyll is one of the dominant pigment which is required for entrapping the sunlight and converting that into the energy in the form of ATP molecules that is being utilized for the production of the organic matters. So the nitrogen is very much important for all the living organism, where the nucleic acids or the proteins or the enzymes or the chlorophyll pigment, they have been made up of this nitrogen. Without this nitrogen, no life would have been existed. However, even we have 78% of the nitrogen concentration in our atmosphere, plants or any of the microorganism, despite a few organisms, nitrogen fixing organism, no other organism has an ability of fixing such atmospheric nitrogen. So how this nitrogen has been utilized by the plants and how they assimilate them, let us study in detail. So before going into proper biological nitrogen fixation, let us study in brief about the nitrogen cycle. As we all know, the nitrogen is one of the important constituent of the atmosphere, where it constitutes around 78%. Still so much amount of nitrogen is present in our atmosphere. Many of the microorganisms or the plants or animals cannot utilize them. This nitrogen can be utilized only in combined form or only when this nitrogen is being fixed. Such nitrogen fixation can happen by three different methods: atmospheric nitrogen fixation, industrial fixation, biological nitrogen fixation. For this process, following steps are involved. One is, first process is ammonification, that is conversion of atmospheric nitrogen into ammonia, and nitrification, conversion of fixed ammonia into nitrites and then into nitrates. Only in the form of nitrates the plants or any other microorganism can assimilate them. Finally, this nitrates which have been assimilated, they undergo the process of denitrification, that is conversion of this nitrogen back into atmospheric nitrogen. Let us study each one of the steps in detail. First, ammonification. The ammonification process can happen due to lightning. During the process of lightning, large amount of energy is released. This energy is being utilized by the nitrogen, where this diatomic nitrogen is being broken down, and this nitrogen combines with oxygen to produce nitrous oxide. This nitrous oxide combines with water and they get landed into the soil. That is one method of the process of atmospheric nitrogen fixation. Then other methods are by biological methods, that is there are few microorganisms. It can be either a free living bacteria or a symbiotic bacteria. It need not have to be always bacteria. It can also be certain blue-green algae, for example Anabaena, nostoc, or any other blue-green algae having heterocyst. They have the ability of fixing the atmospheric nitrogen. Apart from this blue-green algae, there are also certain actinomycetes which have the ability of fixing the atmospheric nitrogen. Say for example Frankia. The microorganism can be a symbiotic or a non-symbiotic bacteria or few blue-green algae, and some of the actinomycetes, they have ability of fixing those atmospheric nitrogen. The atmospheric nitrogen is converted or reduced to ammonia with the presence of an enzyme called as nitrogenase. Nitrogenase is an enzyme which has been associated with the reduction of nitrogen into ammonia. Once the ammonia has been produced at lower pH, slightly acidic pH, this ammonia gets converted into ammonium. So this process of converting the atmospheric nitrogen into ammonium with the help of few nitrogen fixing soil microorganism, the process is called as ammonification. Later, this ammonia, once it reaches the soil, ammonium as such cannot be utilized by the plants. So this ammonia is being converted or oxidized into nitrites by few of the nitrifying bacteria. Say for example Nitrosomonas, Nitrococcus. These are some of the bacteria which are involved in conversion of ammonium into nitrites. nitrites as such, even the plants cannot assimilate. Hence, these nitrites are converted into nitrates by few of the nitrifying bacteria, say for example nitrobacter. These nitrates which are produced or fixed in the soil, such nitrates can be utilized by the plants. The plants can utilize or the plants can assimilate such nitrates. They can assimilate nitrates and use that nitrates for the production of various amino acids, proteins, enzymes, nucleotides, or the nucleic acids, as well as the chlorophyll pigment production. Everything happens through the process of assimilation. Once they have been assimilated, has these plants or producer, these producers are consumed by consumers, and also once the plants or animals die, the dead and decay matters of these plants and animals remains, that is the proteins which are present in these dead and decaying matters are decomposed by aerobic or anaerobic bacteria or fungi. That is, decomposer decomposes these proteins into simpler amino acids, and again these amino acids, or by the process of ammonification, they get converted into ammonium and the cycle continues. Apart from this, there are also some of the microorganisms inside the soil where they have an ability of converting this nitrates back, or they have ability to reduce this nitrates, to convert this nitrates back into the atmospheric nitrogen. Those group of bacteria are called as denitrifying bacteria. The process of conversion of this nitrates back into atmospheric nitrogen is called as denitrification. Some of the microorganisms which are involved in this denitrification are Thiobacillus denitrificans, Pseudomonas denitrificans, Micrococcus denitrificans. These are some of the bacteria which are involved in conversion of this nitrates back into atmospheric nitrogen. So this is about the nitrogen cycle. So once again I will just highlight. Even though nitrogen is the dominant gas present in our atmosphere, it cannot be assimilated directly because this nitrogen is one of the inert gas, because made up of two atoms and bounded by triple bond. Because of this triple bond, they are found to be less active. So only in the combined form this nitrogen can be assimilated by the living organism. So the nitrogen fixation takes place by following steps: ammonification and nitrification. Ammonification can happen by few of the nitrogen fixing bacteria which are present in the soil. They can be either a symbiotic or a free living bacteria which are present in the soil. They have the ability of producing an enzyme called as nitrogenase. This nitrogenase helps in conversion or the reduction of this nitrogen into ammonia. This process is called as ammonification. This ammonia as such cannot be utilized by the plants. Hence, it has to undergo a process of nitrification, wherein which the ammonium gets converted into nitrate, and later this nitrites are converted into nitrates by few of the soil nitrifying bacteria, where this nitrates can be utilized or assimilated by plants, and from plants they can be assimilated by animals or the consumers. Once these producers and consumers die, the dead and decay matters which contain large amount of proteins, they can be decomposed by the decomposers and again the nitrogen is back into soil. The same way, this the nitrates can also undergo a process of denitrification, that is the process of conversion of this nitrate back into atmospheric nitrogen by few of the denitrifying bacteria. Say for example, Thiobacillus denitrificans, Pseudomonas denitrificans, Micrococcus denitrificans, etc. So this is about the nitrogen cycle. Now, let us study about the different methods of nitrogen fixation. As I was mentioning before, the nitrogen fixation can happen by three different methods. We can call it as physical, chemical, and biological nitrogen fixation. First, physical nitrogen fixation, which happen in atmosphere. This is due to the lightning. During the process of lightning, large amount of energy is released. This energy is being utilized by diatomic nitrogen, where the bond between the nitrogen breaks. This atom gets combined with the oxygen present in an atmosphere and gets converted into nitrogen oxides. These nitrogen oxide dissolves in rain and gets converted into nitrates. These nitrates, once it reaches the soil, the plants can utilize them. That is one method of nitrogen fixation. Around 5 to 8% of nitrogen fixation happen by this method when the lightning takes place. Next, industrial nitrogen fixation. This kind of conversion of nitrogen into ammonia is being done in industries for the production of fertilizers. The process is called as Haber-Bosch process. In this method, the nitrogen is being combined with hydrogen with the temperature of around 400 to 500 degrees Celsius at very high pressure of 150 to 250 atmospheric pressure. The nitrogen gets converted into ammonia. Later, this ammonia is being utilized for the process of production of various inorganic fertilizers. And we have biological nitrogen fixation, which happen inside the soil by the involvement of various diazotropes. Diazotropes are the group of bacteria or some actinomycetes or some blue-green algae which can assimilate the nitrogen gas as such. They convert this nitrogen into ammonia in the presence of an enzyme called as nitrogenase, and also this is highly energy requiring process which require ATP molecules for this process. So in biological process, various diazotropes are involved, and with the help of an enzyme nitrogenase, the nitrogen gets converted into ammonia. This is about biological nitrogen fixation. So now, I was discussing about the biological nitrogen fixation. Now, let us study the different microorganisms which are involved in nitrogen fixation. Nitrogen fixing microorganisms, as I was mentioning, they are commonly called as diazotropes. Diazotropes, once again I will repeat, these are the group of microorganisms which can assimilate atmospheric nitrogen, convert them into ammonia, and later they can utilize this for their purpose. Or these are the group of organisms which can fix the atmospheric nitrogen. Those are called as diazotropes. Diazotropes can be classified into heterotropes and photoautotropes based on the presence or absence of chlorophyll pigment in them. we can classify them as heterotrophs or photoautotrophs. Heterotrophs are usually dependent or they cannot synthesize their own food. These heterotrophs can be classified into two kinds, free living and symbiotic. Free living heterotrophs, again we can classify them into based on the oxygen utilizing by these group of organisms. They can be classified into aerobic, facultative, anaerobic. Aerobic bacteria which are free living, which are heterotrophic diazotrophs, are Azotobacter, Azomonas, Bajrankia, Rhizobium, Azospirillum, etc. Facultative anaerobes. These are the group of organisms which can live in presence or absence of oxygen. They are called as facultative anaerobes. Bacillus and Klebsiella species has an ability of fixing the atmospheric nitrogen. Likewise, we have anaerobic bacteria, Clostridium, Desulfovibrio and Methanobacterium species. These are some of the free living heterotrophs which are having the ability to fix the atmospheric nitrogen. Likewise, there are symbionts, symbiotic bacteria which are heterotrophic in nature. Symbiotic bacteria which can fix the atmospheric nitrogen can be classified into two kinds. They can be nodule forming or non-nodule forming. Nodule forming bacteria are the one which forms a nodule or a knot-like or some gall-like structure on the roots. Inside this nodules, the nitrogen fixation takes place. Those are called as nodule forming bacteria. Nodule forming bacteria can be legume or non-legume based on the plants, the family of the plants they infect. There are some group of bacteria, say for example Rhizobium, the group of Rhizobium, which have an ability of forming a root nodule and can fix the atmospheric nitrogen in the plants belonging to the family Leguminosae, Fabaceae, groundnuts, peas, soya beans. All this are the group of organism which belongs to the family Leguminosae or Fabaceae. Only in this group of plants they can form a nodule and fix the atmospheric nitrogen. Likewise, you have nodule forming bacteria, say for example in non-legume, example Frankia, Anabina, Nostoc, etc. Likewise, we have photoautotrophs. Diazotrophs, once again I will repeat, can be classified into heterotrophic and photoautotropic. Heterotrophs, they depends on other plants or other source for their food, whereas photoautotrophs, they can synthesize their own food. These photoautotrophs can be classified again into two kinds, whether they are free living or they are symbiotic. Free living photoautotrophs can be classified into a cyanobacteria or a blue-green algae or other bacterium. Cyanobacteria group, which includes Anabina, Nostoc, Cytonema, Glyocapsa, Rivularia, Limbia, etc. Likewise, you have other bacteria like Chlorobium species, Chromarium species, Rhodopseudomonas species, Rhodospirillum species. These bacteria also has an ability of fixing the atmospheric nitrogen apart from photosynthesis also. Likewise, you have even photoautotrophs are also found to be symbiotic, where the symbiotic ones can be classified into non-nodule forming, nodule forming. Nodule forming, we have Nostoc, and we have non-nodule forming Anabina, Halosiphon, and even some other species of nostoc. Even Anabina cycadacetum also can be found in the coralloid roots of cycads. Even that can also be called as a nodule forming bacteria. So these are some of the example of nitrogen fixing microorganism. Kindly remember, try to. Now let us study about the symbiotic nitrogen fixation. Taking the example of Rhizobium, let us study this in detail. This Rhizobium is a group of soil bacteria which are gram-negative, aerobic, and in presence of the host, in presence of proper host, they have an ability of fixing the atmospheric nitrogen. This Rhizobium species, each species of Rhizobium, they are species specific, that is, they are found to be associated with a leguminous plant which are specific. They are found to be highly specific for the process of symbiotic association, and also they have ability of producing the root nodules. Root nodules are produced in the leguminous plants, and they can fix the atmospheric nitrogen only inside the root nodules, because root nodule formation is highly important for the process of nitrogenase activity. Nitrogenase is an enzyme which can fix the nitrogen or convert or reduce the nitrogen into ammonia. This nitrogenase is highly essential, and this nitrogenase is highly active only in the presence, only in the absence of, that you have to remember, only in the absence of oxygen the nitrogenase is active. So if the oxygen concentration increases, the nitrogenase activities stop. The operon, NIF operon, is a cluster of genes which has the ability of fixing the atmospheric nitrogen by the production of an enzyme nitrogenase. The NIF operon gets switched off if the concentration of oxygen increases. There are certain proteins are also produced by this rhizobium called as leg hemoglobin. These are proteins which are also called as the oxygen scavengers. These proteins are produced only in association of the rhizobium inside the leguminous plants. So this leg hemoglobins which are produced by the rhizobium in association with the leguminous plants, they scavenge, they see to that the oxygen concentration is limited. So this leg hemoglobin is also very much essential for the process of nitrogen fixation, so which happen inside the root nodules. And also there are certain genes called as nodulation genes. This rhizobium also have an ability of producing a factor called as nod factor, which we are going to study in detail about these genes which are involved, what is the mechanism of action, or how actually the regulation of this operon of nod regulates. All those we will be studying in our future slide. Now let us remember that there are certain genes called as nodulation genes, or also called as nod genes, and also nitrogen fixation genes or the NIF genes are present inside the rhizobium. Symbiotic associated rhizobium, you can find such gene inside a plasmid called as symplasmid, or it is also called as symbiotic plasmid. In this symbiotic plasmid, we can find a peron or a cluster of genes which are responsible for nitrogenase production called as NIF genes, as well as we have a nod genes which are responsible for the formation of nodule in leguminous plants. And in this particular slide also I have given few example of a group of rhizobium. Say for example, peas. In peas, the rhizobium species called as Rhizobium leguminosarum, a biovar, that is we see. This biovar is nothing but the variety of leguminosarum. So likewise we have bean, Rhizobium leguminosarum biovar fascioli. So this indicates that this rhizobium, they are species specific and also strain specific or variety specific. So it is not that the same rhizobium is present in bean or in cowpea or in horse gram or in peas or in clover or in soybean. So in each host the rhizobium, they are specific. There are also there are certain binding proteins on the surface of the root hair of these leguminous plants, and also they have an ability of producing certain signaling molecules. These signaling molecules in the form of flavonoids, these flavonoids or signaling molecules are specific for particular plant and particular strains or the species or the varieties of the rhizobium. So this indicates that the rhizobium are species specific for the process of symbiotic association. Likewise, you have mesorhizobium and you have cynorhizobium, bradyrhizobium, and also azorhizobium. All these are commonly called as rhizobium species. So these are some of the examples of rhizobium which are found in a particular host plant, especially the leguminous plants. Please try to remember these species as well as the host. Now, this is a slide which is just a glimpse on how actually the nitrogen fixation happen in the rhizobium. So this in detail we are going to study in my future slides. So here how actually the nitrogen fixation happen inside the rhizobium. The atmospheric nitrogen can be assimilated by such diazotropes like this symbiotic rhizobium. This atmospheric nitrogen is being absorbed by the rhizobium, and there are certain enzymes, as I was mentioning before, an enzyme called as nitrogenase. Concentrate on the image please. Nitrogenase enzyme. This nitrogenase enzyme involves in the reduction, as well as there are also certain enzymes called as hydrogenase. These are the enzymes. Hydrogenase are the enzyme which reduces, which are actually which reduces the nitrogen into ammonia. The ammonia at slightly acidic pH gets converted into stable ammonium. This stable ammonium later it's been transported or translocated into the plant. Since I am talking about the symbiotic association over here, inside the nodule all such processes are happening. Inside the nodules, the rhizobium, which is an aerobic gram negative bacteria which present inside the nodules, they absorb the atmospheric nitrogen, convert this atmospheric nitrogen into ammonium with the presence of an enzyme nitrogenase, and for this process large amount of ATP is also involved. This ATP is being synthesized inside the rhizobium by utilizing the carbohydrate source from that of a host. This ATP molecules or large amount of energy and the nitrogenase enzyme is involved in conversion of this atmospheric nitrogen into ammonia, and also remember hydrogenase is also involved. And apart from that, there is a protein, as I was mentioning, protein called as leg hemoglobin. This leg hemoglobin can be synthesized only when the rhizobium is present inside the nodule, only in association with leguminous plants. They can be produced. So they are called as leg hemoglobin. They acts like a oxygen buffer or oxygen scavengers. They scavenge the oxygen so that proper concentration of oxygen is being maintained inside the cell of the rhizobium, so that the nitrogenase activity is being activated. So these each one of this step we are going to study in detail in this particular presentation, and also in my future presentations. So now As I was mentioning, taking an example of symbiotic bacteria called as Rhizobium, I said that Rhizobium can fix the atmospheric nitrogen only inside the root of leguminous plants by the formation of nodule. So in this slide, let us study how these nodules are produced. So the different steps which are involved in the formation of nodules goes on like this. First is attachment of the rhizobium onto the surface of the root hair. Before attachment, it is recognition. That is, rhizobium has to recognize proper host. The rhizobium cannot go and infect any roots of any plant and they can form a nodule. It is not like that. They are very much species specific. They get the signaling molecules from the plants. Plants in their rhizosphere region, in their what is this rhizosphere? It is a region or the soil which is surrounding the root system is called as rhizosphere, and the microorganism which are present in such soil is called as rhizosphere microorganism. The roots of the leguminous plants produces certain signaling molecules called as flavonoids. These flavonoids acts like a signaling molecules for this rhizobium. This rhizobium gets attracted to such flavonoids and they start with the infection. So the first step in nodule formation is recognition of proper host. The second step is attachment of rhizobium onto the root hair. Once they attach, how actually they attach? Let us study all those later. The different steps which are involved in root nodule formation is recognition, attachment, penetration, formation of infection thread, and entry of this bacteria inside the host cell, and bacteria gets transformed into bacterioids inside the root cell. The root cell enlarges and the root cell enlarges and they form a nodule. These are the different steps which are involved. Once again I will repeat, the different steps which are involved are: first one, recognition; the second, attachment of rhizobium onto the root hair; infection, that is the formation of infection thread through which the bacteria can enter into the root cell. Once they enter inside the root cell, they get changed into bacterioids. Bacterial cell enlarges, divide and redivide, and they form a structure called as nodule. These are the different steps which are involved in the process of nodulation. Now let us study each one of the step in detail. As I was mentioning, the first step is recognition. If the plant produces appropriate signaling molecule called as flavonoid, once they are exudated, that is, release of such substances into the soil, the process is called as exudation. Once it has been exudated, the microorganism, especially the rhizobium, which are present in the rhizosphere soil, they recognize such flavonoid molecules and they start to attach onto the surface of the root hair. How they attach? These rhizobium has certain proteins called as Rick adhesin. Remember, they have a special proteins called as Rick adhesin. This Rick adhesin helps in the attachment of the rhizobium onto the surface of the root hair. This Rick adhesin is a calcium binding protein. It's a calcium binding protein. With the help of this Rick adhesin, they gets attached onto the surface of the root hair. Root hair also has got certain attachment site, or they have a receptors. Only onto their receptors made up of a special carbohydrates called as lectins. On this receptors, rhizobium come and bind onto the surface of the root hair. At that particular point of time, rhizobium also has an ability of producing a substance called as nod factor. Please remember, they have an ability of producing a substance called as nod factor. This nod factor triggers, or it sends the message to the nucleus of the cell of the host to transcribe the proteins or the substances which are required for the process of the infection and for the process of colonizing, or for the process of the formation of nodules. So these nod factor, they stimulates the nucleus of the host to transcribe for the production of proteins which are required for the process of the formation of infection thread, as well as for the process of the formation of bacteroids, or for the process of the formation of nodules. All those about this dot factors and all we will study in detail little later. Now, once they attach, they have to infect. During the process of just before the infection, this bacteria, they produces nod factors. These nod factors, as I was mentioning, they help in the process of the formation of a cellulosic tube. These cellulosic tube are produced by that of a host. Through this cellulosic tube, the infection, or it's also called as infection thread, through this tube, this rhizobium enters. They divide, redivide in this cellulosic tube. They enter into the cortical region of the host root, and once they enter inside the cell of the host, the plasma membrane of the host gets accumulated even onto the surface of this rhizobium cell, and that process is such structure which has been formed on the surface of the host, the layer which has been formed due to the accumulation of the plasma membrane or the symbiosome which has been produced by the plant. Such deformed structures, they are called as bacteroids. Once this happen, the nod factor also stimulate the host cell to undergo a process called as nodule organogenesis. This is nothing but the cortical cell, again they acquire the characteristic feature of dividing. These cells, cortical cells, divide or redivide, and also sometimes they undergo process of hypertrophy or hyperplasia, as I had explained in my previous video lecture. So this is all due to the phytohormones, auxins and cytokinins, which are majorly involved here. They involved in the process of division of the cells and to form a mass of cells which are called as nodules. Once the nodules have been formed, once the symbiosome has been accumulated on the surface of the rhizobium, then the nitrogen fixation starts. So these are the different steps which are involved in the process of nodule formation. Once again, the different steps are recognition, attachment, formation of infection thread, and bacteroid formation, and the enlargement of root cells, and finally for the formation of root nodules. So again, this diagrammatic representation may also be useful to understand better. So the nitrogen fixation, or the different steps which are involved in the formation of nodules in leguminous plants of Rhizobium species, goes on like this. The first step is, as I mentioned earlier, a recognition of a proper host. That is, uh, the plant releases certain signaling molecules called as flavonoids. These flavonoids, if it is appropriate, the host, so Rhizobium cell also produces certain signaling molecules called as nod factor. Because of the production of this nod factor, the infection starts. Before infection, they attach with the help of a special protein called as Rick addison. Rick addison, they are actually calcium binding protein, and also there are certain lectins which are produced by the plants. They are actually the receptors which are present on the root hair. On this receptor, the Rhizobium, with the help of Rick addison, go and bind, and finally they produce the factor or certain substance called as nod factor. These nod factor stimulate the host to transcribe cellulosic tube. Through this cellulosic tube, the bacteria enter. After curling the root hair, get curled, and it forms the infection. Once the infection has happened, the cell, the Rhizobium, penetrate inside or passes through the cellulosic tube inside the cortical cell, and finally this plant cell also produces various phytohormones, and also the things which are required for the process of nodule formation. The nodule organogenesis happen, where the cells in the cortical region, they get again they get the ability of dividing and redividing, which leads to the increasing the volume of the cell or the number of the cell. And this such bacteria, Rhizobium, enters into such cell, and at the same time they get accumulated with the membrane which has been secreted by the host, that's called as symbiosome. Once the symbiosome has been secreted, this bacteria loses its morphology. They become polymorphic. As we know, they are of different shape, and once the symbiosome has been secreted on them, the nitrogen fixation starts. So these are the different steps which are involved for the process of nodulation.